database of intrinsically disordered proteins. So in the previous webinar, we described how to perform a basic search in DisProt and how to explore the home page of the database, and also how to use the related documentation such as the help, about, biocuration, and release notes pages. So today we are going to go a bit more in detail about the features of a DisProt entry. We are going to see a specific use case and also how to use the browse page for specific, more complex searches, how to download an output of a search. So to start with exploring a specific entry in DisProt, you can look for the name of a protein or a specific identifier if you know it here in the search in DisProt. So what we are looking for now is a protein related to autophagy. Okay, here it is. We are going to explore autophagy-related protein 13. And this protein, as you have seen, we discussed the previous time that for each one of the entries in DisProt, there are details such as the organism, the sequence length, and disorder content, but also cross-references to other resources such as UniProt, AlphaFold, MobiDB. If a protein is part of one or more specific datasets in DisProt, we are going to see it here. As you can see, this protein is part of the autophagy-related protein dataset. And then you can see when was the protein last updated in DisProt. So what we can appreciate in this page is that each DisProt entry is composed by two main sections. One is on the top of the page with the information about the protein and the feature viewer, and then the second part of the page with the annotations provided by biocurators of DisProt. So let's start looking at the feature viewer now. As you can see from the feature viewer, this is the annotated disordered region of ATG13. This is the corresponding color and the structural state in disorder for this region. As you can see, this completely matches what AlphaFold predicts for this protein. You can see these regions in yellow and orange that correspond to a low or very low PLDDT, lower than 70 or 50. This corresponds to intrinsically disordered regions, and indeed the second half of the protein is disordered. And we can also see that we have other type of annotations related to this intrinsically disordered region, such as the molecular function, cellular component, biological process from gene ontology. And if you look here and you scroll over this bar on the feature viewer, we're going to see which is the length of the fragment. So from residue 268 to 738, the region is disordered. The same way, if we expand these areas of the feature viewer, we can see which are the other annotations provided for this protein. So this protein, for example, at least part of the ID region, is involved in the protein binding and lipid binding. And you can see here which are the regions, the annotations that are involved in protein binding. So for example, this with the specific identifier for the region, the name of the curator, the fragment, the method used, which is immunoblotting, and the reference of the paper describing that this protein is involved, this region, particular region, is involved in the binding of a protein partner. The same way, we have the regions inside the main disordered region that are involved in protein binding. For example, here, it's a region 359, 389 that was assessed by isothermal titration calorimetry. Again, we have the reference of the paper describing this region. But we can also see that we have other type of binding, like for example, lipid binding that we can see here and here. So these are from gene ontology. These are three domains annotated in gene ontology: molecular function, cellular component, and biological process. For the cellular component, you can see that this protein is part of specific complexes. For the biological processes, you can see what this protein is involved into. So it's involved in the regulation of cellular localization. You can see here, this is the region, and it was assessed by fluorescence microscopy. You can also see, being an autophagy-related protein, it's involved in the positive regulation of autophagy. For example, here, it was assessed by mutant phenotype evidence that this region plays a crucial role in autophagy. Here, you're able to see that there are the domains such as PFAM, Gene3D, and you can see indeed that this is the structure region of the protein. Finally, you will be able to appreciate the prediction of AlphaFold with the model confidence for each fragment. For example, this is a fragment predicted as highly structured with a very high PLDDT. This region is the one that is predicted as disordered. If you look at the second part of the entry, you will be able to explore all the annotations provided by our biocurators. The annotations before being public are passed by of the reviewers, DisProt, so expert biocurators, and they become public with the next available release of the database. We have releases every 6 months, so in June and December. So you can see here that we have annotations for the structural state, the disordered structural state of the protein, indeed corresponding to this region. And these are provided by different papers and also different methods. As you can see, the region is the same, just an amino acid, shorter because it depends on the construct used by the authors, but we have two different papers, for example, here, and the same method. Both these papers are describing this region as disordered. But you can also see here another paper analyzing a shorter region of the C-terminus of ATG13 with far UV circular dichroism. Again, they demonstrate that the region is disordered. What you can do then is explore molecular functions that are associated with this protein. So, as we have seen before, protein binding, but you can also see, if it's involved in protein binding, which is the uniprot identifier of the corresponding partner that binds that specific region of ATG13. Again, you can also see the cellular components annotations and the biological processes that are annotated for this protein. So, what can you do if you want to perform a more complex search in DisProt? Instead of using the home page where you can simply look for the name of a protein or a specific DisProt identifier, you can use the browse page. The browse page, as you can see, lists all the entries in DisProt, the entries that are public 400, and you can explore them by looking at the different pages. But of course, you might be interested in seeing more details than those that are shown here. The basic details that we show are the DisProt identifier, uniprot accession number, the protein name, the organism, the sequence length, and the disorder content. But we can also customize the columns. So here you see the columns that are already selected for visualization in blue, but you can also choose to show other information, such as the annotated evidence codes, meaning the experimental method used to determine and describe a specific disordered region or one of its terms, and for example, the annotated terms. The annotated terms are the terms from the Intrinsically Disordered Proteins ontology or gene ontology that describe, for example, the structural state of a protein, the structural transition, the functions, as biological process, molecular function, cellular component, or the functions specific to an intrinsically disordered protein or regions that are defined in the ontology page DisProt. We are now going to explore this page too. So you can see here now that if we customize the columns this way, we will have more information. So for each protein, for example, this one, we are going to see that we just have an annotation of a disordered state, and that this annotation is provided by an X-ray crystallography experiment. For this protein, for example, we do not only have an annotation of structural state disorder, but we also have a transition annotated. Indeed, we have a region that changes state from disorder to order and is involved in a biological process, cytolysis. There are indeed more methods here used to describe these structural states, transitions, and functions. So what you can do, you can customize the columns as we have seen. There are also other information you can add as the NCBI taxon. You already have the organism here, but you can add the NCBI taxon too. You can add the UNIREF 150 or 90. Let's assume that you want to look for something more specific. How can you do it? We have a specific select search field here. These are all the fields that you can look for in DisProt. We have the protein name, the UniProt or DisProt identifier, the organism, the dataset, taxons, UniRef, but also you can look for a specific curator annotations, or what is more interesting is a specific ontology term from the IDP ontology, the gene ontology, or the evidence and conclusion ontology. As you have already seen, intrinsically disordered protein ontology and gene ontology provides annotation about functions of a specific intrinsically disordered region. The evidence and conclusion ontology is what we use, the ontology we use to annotate the experimental method used to describe a specific region or one of its functions in a publication. What you can do if you are not familiar with a term of the ontologies annotated in DisProt, you can explore the ontology page. So see, DisProt relies on 3 different ontologies: the intrinsically disordered protein ontology, the gene ontology, and the evidence ontology. For these proteins ontology, you see that we have 3 different branches, that is the structural state, so the disorder, or one of the other states, such as pre-molten globule or molten globule, transitions, and functions. You can explore the intrinsically disordered proteins ontology here. Let's say that you're interested in a specific function associated with an intrinsically disordered region. You can look here and you can see the functions that specifically pertain to intrinsically disordered proteins and regions. For example, a very common functions of intrinsically disordered proteins is the flexible linker spacer, which is a disordered region that connects and provides separation between 2 domains that are structured. But you can also look for the gene ontology annotations here. You can explore the gene ontology by using the links that redirect to Amigo or QuickGo to explore gene ontology. And here you can look for all the terms that we use in DisProt from the evidence and conclusion ontology. We used mainly experimental terms, so terms that describe a specific experiment performed in a paper. So let's say that we want to look for a specific function from the gene ontology. You can look for the identifier, which is more difficult because it's a series of numbers, but you can also look for a specific term name. So if we want to look for one function, we are going to see more informations here. For example, we have the proteins that have an annotation related to regulation of autophagy. for example, the von Hippel-Lindau tumor suppressor that is indeed involved in autophagy. We have also Becklin, again involved in autophagy, and other proteins that have annotations associated with this term. For example, the autophagy-related protein 13 that we just described a while ago. So if you find some information that are useful for your research or that you're interested in DisProt, you may want to download them. So how you can do this? You can click on the download selected that you see here. So here we retrieved 4 entries, okay, that correspond to this term. What you can do is choose the type of aspect that you want to download. What I recommend is all. Please download every aspect annotated in each one of these entries. Otherwise, you can choose to download just a specific aspect. Let's say maybe the structural state or a disorder function from the IDPO, or one of the specific domains annotated from gene ontology, the biological process, the molecular function. Once you have done choosing the aspect you want to download, you can choose the type of data that you want to be included in the output file. So the regions or the consensus for each region. Again, the default setting is region. You can then select the format because we have 4 different formats available: JSON file, TSV, and FASTA. The GAP format is a specific format related to gene ontology. So this is one way for you to download the output of a search in DisProt. But maybe you're interested in a specific protein. What you can do is open the corresponding page and download the information annotated only for that protein. Again, you can choose the type of aspect you want to download. You can select all the annotated aspects, or you can choose among one of the aspects annotated, which is the structural state, disorder function, transition, and interaction partner for this specific protein. Once again, you will be able to choose the type of data and select the format for download. Finally, we also have a dedicated page where you can perform download on DisProt, maybe on a specific release of the database. So this is the current release, the June release of DisProt. You could choose also a previous release, but let's assume that you're interested in the current one. You can download all the data about this release here. What you can also do is including ambiguous or obsolete annotations. I recommend not including ambiguous and obsolete annotations. Again, you can choose from the file formats JSON, TSV, FASTA, and GAF. Maybe you could be interested in a specific aspect. We have seen that we have the transitions, the state, the functions from the Intrinsically Disordered Protein Ontology, or the gene ontology functions. So you could choose molecular function and download the associated information here. Then you could also be interested in downloading the information about a specific dataset. So far we have in DisProt 5 datasets that are these: extracellular matrix proteins, unicellular toxins, antitoxins, viral proteins, cancer-related proteins, autophagy-related proteins. So if you're interested in downloading all the proteins in the autophagy-related proteins, which was released in June 2022, you can select it and you can click for download on a specific file format. If you're interested instead on the IDP ontology terms, you can select the version of the ontology and you can download a JSON or an OBO file corresponding to that version of the ontology you want to download. If you're interested in the dataset, what you can do is also exploring the datasets directly from the homepage, clicking on the dataset. Here you will see for the autophagy-related protein dataset that there are currently 101 proteins that are part of the dataset. You can directly download it here, maybe excluding ambiguous and obsolete entries. You will click on download selected. It's now 100 selected entries, removing the ambiguous, and you can click on the TSV file or on the FASTA file corresponding to your dataset of interest. So this is all for what concerns the use of DisProt, explored in the browse page, and the main features associated with DisProt entry. And thank you for your attention.